you are going to learn all these type of animals in the world. Animals, mainly extant species. Extant species in the sense those species not yet extinct. Extinct means disappeared. So we are going to learn those animals living today, starting from very minute animals and up to vertebrate and mammals. And these type of animals, animals in relation to the plants, and animals in relation to different ecological systems, as well as animals related to even domestic environments and all these. So if we take now the actual learning outcomes of this particular course module, that is 14703, what you are going to gain at the end of this unit, what you are going to gain, or what you are going to achieve, or what you are going to be able to do it after following this course. First is, you should be able to explain the basic principles of animal taxonomy. So what is animal taxonomy? We'll learn what is animal taxonomy. So at the end of this semester, following this course unit, you will learn how to explain basic principles of animal taxonomy. Basic principles. What are the principles based on animal taxonomy? Then you will be able to explain again the architectural pattern or the body pattern and evolutionary trends in the animal kingdom or kingdom Animalia. So throughout this course unit, you will learn only the features of the animals, that is the animals of the kingdom Animalia. Kingdom Animalia, how they have related different animals, how they relate their evolutionary trends, and how their body patterns or architectural patterns related to each other. You should be able to explain. Then again, you should be able to describe. Describe means again type of explanation. Explanation. The major characteristic features. Major, not all the characters. Major characteristic features and diversity of animal taxon. So what is animal taxon? Then you are going to learn what is animal taxon. Individual animal, their characteristic features. And not only just characteristic features, major characteristic features, three important words you are going to describe correctly at the end of this course unit and their diversity. Then you will again able to discuss giving examples and detail the adaptive radiation. So what are adaptive radiations of selected animal groups? Adaptive radiation means if we take a single animal, it has specific characteristic features, specific characteristic features. But there may be another animal do have another specific characteristics, but giving similarity to the previous animal. So if we take the first animal here and the second animal here, there are some relations, similar features. Likewise, if we take the third animal here, still there are kind of similarities. And if you take another animal here to the first type of animal, you find again further similarities. Likewise, you take different number of individuals here, individual animals, you find some common features or similar characteristics. It is like radially arrangement with the main features of the first animal or animal group we are going to describe. So that you should be able to describe and discuss the adaptive radiation of only selected animal groups. So there is a vast animal group, so you can't complete all those animal groups within this course unit, so that you are going to learn selected animal groups adaptive radiation. So those are the learning outcomes. Learning outcomes we call learning LO. Learning outcomes of this course unit. Usually the learning outcomes we refer as we are the expected learning out. Expected. And we call it as intended. Intended learning outcomes, so that we refer it as intended learning outcomes or ILOs of ZOOL 12703. Okay. Now you are very clear about what are ILOs or intended learning outcomes of ZOOL 12703. And what are the things you will be having or you will be explained at the end of learning this course unit. We'll try to do maximum out of it, the course content. And there, out of the several recommended textbooks, there is a famous book written by Hickman with different authors. And today you are going to learn, those are the series. Now, first 2 lecture hours, that is today and next Thursday, we are going to learn the introductory part to the animal diversity. Introductory part to the animal diversity. And very beginning, before we come up to the exact animal phyla or the phyla of the animal kingdom, we'll see what are the principles based on organism classification. What are the principles based on classification of organisms? It is not only animals, all living organisms. And the types of different classification used in old days. And the classification or classification system we are using today. That is linear classification and how it is improved with after several years of time. So the introduction to the animal diversity start with principles of classification or principles of classification of organisms and the types of classification of organisms and the linear classification system. All these explained in the chapter 3 and chapter 4 of the textbook and actually referring the animal diversity. Animal diversity is a little in-depth or very much theory-based course unit, but which is interesting if you take interest of learning. So the animal diversity refers mainly to animal evolution. It is not animal evolution, it refers to animal diversity referring to animal evolution. While animal evolution only produce different types of animals into the earth. Different types of animals into the earth. Different shapes, different types, different sizes, like this. So the animal diversity exists since there was an evolution. So these animals in animal diversity may be vertebrate type of animals or invertebrates and very tiny ones or arthropods, crustaceans, poriferans, sponges, or cnidarians, ctenophores. Likewise, any different type of animals, they add to the earth in a time series. So more and more animals adding to the earth will increase the diversity. Increase the diversity among the animals. So we are referring to animal diversity. Therefore, animal diversity refers to animal evolution. So animal evolution initiated expecting they evolved from, or life began from the ocean. Life began from the ocean. So it is the photon initially. So you have to imagine now there must be some kind of an organism similar to animals remain in water. That is the origin, water. So that particular thing initiated, that is beginning of the animal life, that is similar to animals in this much amount of years of time before. 600 million years ago only, this animal origin took place in the ocean. So if we are consider how this type of animal origin from the ocean or water, it is not the animal you can explain as it is an animal. It is completely different shape, different arrangement, body arrangement, and even you cannot tell that it is similar to the animal. You cannot explain even that it is an animal. So that particular creature actually do not similar to any living organism today. Any living organism living today. It is somewhat different. Or that particular creature originated in ocean 600 million years ago does not show any similarity to the animal living today. Somewhat different. Okay. Now you can see the image here. This is the kind of creature that initiated the whole life of living creatures today. It is like a flood, and you can see a single nucleus. And you can see it doesn't have a shape, but you can see from this particular creature, which is not similar to any living organism, you can find it here. Plants, different. Invertebrates, different. Then vertebrates, different. And this one is completely different. Okay, this is how the evolution. It is the animal here in this part. Animal evolution originated from the ocean 600 million years ago from a creature that do not resemble any living organism today. Okay. And if we take here, I said any animal evolution, part of the whole evolution originated from the ocean 600 million years ago from a different creature, not similar to nowadays, that included plants and animals of living today. But if we take animal alone, if we take animals alone, the animal origin, according to most of the scientists, believe originated from a colonial protist. Protist in the kind of animal protista. Nowadays, we consider as kingdom protista. And organism of the kingdom protista do have category of organisms that shows individual organisms attached to each other, we call it as a colony. Individual animals attached to each other forming a colony, we call colony type. Colony type protista. Single protista here, this type of cell body with the flagellum and the nucleus. And you can see this type of organism with the flagellum forming a colony like this. Colonial protist named as choanoflagellate. So this is the typical description of a coelom that expect from where the animals originate. That expected, according to many scientists, how the animals originate. So your answer for how the animals originated, animals living today originated from a colonial protista, that is coelom protist organism found in ocean. Found in ocean. Here you can see this particular organism lived in ocean. Blue color background, ocean, but it is not simply or freely moving. This is the colony form, flagellated protista. Flagellated means having a flagella. Flagella means a simple thread-like structure that they move to create water movement. Create water movement. Create water movement. Okay. So this particular organism live in the ocean. You can see these organisms live in the ocean attached to a substrate surface. Attached to a surface with a stalk-like structure. Stalk-like structure attached to the substrate, we call it as sessile, so that they are unable to move, attached to a surface. Since they are unable to move, how can they obtain their nutrition? And how can they obtain their nutrition? How can they escape from predators? And how can they meet their mates? All those things they create by this water movement, creating a water current by the movement of this flagellum. So this is the type of organism now we consider as evolved into the present day animals initiated from the phylum Porifera. Phylum Porifera includes sponges. So phylum Porifera is the first animal phylum that we are going to learn in next few days' time. So you will learn from there how the phylum Porifera keep related to this particular individual, that is to make a link, that animals evolved from this kind of individual. So if we take this particular individual organisms attached to each other, it is somewhat like this, attached to the substrate. But if we take a cut across, after a several evolutionary trends, you will find these individual organism, it is an individual cell arranged superficially like this, arranged superficially, and in the middle it is a hollow. That is the most primitive type of animal we can consider evolved from this particular individual. Initially, in the middle it is a hollow, and the cell layer doing different functions, arranged superficial, so that the flagellum exposed to water, so it creates the flagellum to move, capture the food, and the cells grow. But development further happen in individual steps with improved development. You can see some kind of cells arranged similar way here change to a reproductive cells. Reproductive cells means doing a different function only. But previously, all these cells did reproductive function as well. Now you can see when the animal develop further in this category of cell, one category of cell, they differentiate label. Weber differentiation means one cell do one kind of thing, another cell do a different kind of thing. So here you can see ordinary body cells, somatic cells, and some only develop as reproductive cells. And these reproductive cells, you can see, remain only one region, doing only the reproductive function. Rest of the cells develop further, and from here, these cells in the latter stage of development, you can see the cell layer impressed into the hollowed area appeared in here. Hollowed area. Cell layer pressed, depressed. So that you can see further and further depression take place until this cell layer, this area, go and lie just underneath the superficial layer of the cell. So it made first layer and the other is the second layer, two body cell layers. And whereas the area, the cells move interior, form a hollow hill. It form either a mouth or a anus of the adult individual. That is how the animal further development take place with different body layers. From the originate of the animal, that is colonial protista, that is in the kingdom protista, type of flagellated animals, flagellated organism attached to each other forming a colonial protist choanoflagellate. We call proto-animal. Then here you can see just a phylogenetic, development of the individual animals, that is animalia. And here it is the plants, like previously, all these in different. From here is the choanoflagellum. The individual we referred previously. Choanoflagellate. From here upward are the animals. So the importance of choanoflagellate, that is the origin of animals, we call proto-animal. From there, they develop as further animal groups, animal phyla, and up to human development. And here is just elaboration how the choanozoans or choanoflagellate as ancestors to the development further. Here the colonial form of choanoflagellate. Here is the individual organism. This one, you can see they can able to form a cyst to undergo unwanted environmental conditions, and the cyst further develop as asexual ones, asexual reproduction, and some they develop as amoeba type individuals, and some they develop as colonial type. From here, the animal kingdom originated as expected. Here is the two categories. Again, the choanoflagellate. Here is the choanoflagellate, and how the animal group evolved with different groups, fungi, plants, animals. Okay. Now we look at how the animal classification system and the system characteristics and the characteristics of animals based on different categories. So the animal classification. So now we are going back to how we have classified animals for different organisms. So this classification is basically classification of organisms basically did with their anatomical features. Anatomical features means their structural features. Structure. and the physiology, internal structures. Then the classification basically based on the morphology. We call morphological features. Then also the classification based on evolutionary history, how they have evolutionary history, and also how the features of embryonic development and the genetic makeup, genetical. So actually, the animal classification system characterizes based on different features, anatomy, morphology, evolutionary history, how they develop in embryonic or the embryonic development, and how they make up genetic variation. Those are the features nowadays we consider to classify animals in animal kingdom. That is after originated from coelom-flagellated type of individual. So in the very early days, we classified animals. We classified animals basically whether they have red blood cells or whether they don't have red blood cells. Red blood cell. If they have red blood cells, they classified animals into this category. Birds, fishes, whales, then oviparous quadruped creatures and amphibians, then viviparous quadrupeds or mammals. Oviparous means egg laying, viviparous means individual laying, baby born and egg laying. Amphibians, mammals. Those included having red blood cells, animals, and the invertebrates, those who do not have red blood cells in this category. Cephalopods, that is molluscans, crustaceans, arthropods, then insects in the arthropod, then shelled animals like molluscan, and the plant animals we call nidid. That is the old days classification we do not consider nowadays. So that is the history. And if we take here as the taxonomy, remember we said very initially we are going to learn about animal taxonomy. And what is taxonomy? So taxonomy is kind of a biology, that is kind of a zoology, deals with identification and naming of organism, that is taxonomy. But we are learning here animal taxonomy. So the animal taxonomy deals with branch of zoology that deals with identification and naming of animals. So the taxonomy simply you can describe identification and naming, how we are going to name. So naming we refer as nomenclature. Naming is nomenclature. And the systematic classification, we call it as systematic. Classification or grouping, group the animals. We are going to group different animals based on different features. What are the features we are looking for? These are the features: anatomy, morphology, evolutionary history, embryonic development, and genetic makeup. We are going to classify the animals. We call systematic classification. So systematics and the nomenclature go together. That is how we are going to describe individual animal or name individual animal. So the taxonomy is identification and naming the organism, this part. So once we name the individual, we should be able to classify the individual. That is systematics. So the advantages of taxonomy. Why do we do this? Why do we learn animal taxonomy? Why do we learn animal systematics? That is to make easy for description and the study of organism. Okay? Describe and study the organism advantages. Then also it will help to memorizing the individual. So there is a pattern of memorizing individual, otherwise they may go unnoticed. Then also it will improve predictive powers, predictions, improve predictive powers or improve or enhance predictions. Then improve explanatory powers about among organisms' relationships. If we learn the taxonomy, we should be able to give some impression about the relationship between different organisms. Here the animals, relationship between individual animals, that will improve the explanation between two individuals with their relationship. Also, this will help or provides a scientific name, taxonomy. Taxonomy, after learning, ultimately end up with scientific naming. In zoology, we call zoological naming. So the naming or the taxonomy, after learning the taxonomy with naming the individuals, that will help us to identify a particular kind of animal. If we take human, commonly we can refer the human as, in English, man, in Spanish, hombre, in German, bird, in Chinese, ring, Latin, homo, Sinhala, different things. So, but if we can learn one particular category of nomenclature, we can refer these particular humans as only home. Whether it is in England, whether it is in Spain or German, China or in Sri Lanka, we refer only by single word home. Otherwise, it is going to be a confusion. That is only human, but if it is an unknown individual or unknown animal, if we give different names, common names, same animal, if we found in a different country, we are unable to make it clear whether it is the same animal referring to that particular different type of names. So it is the accepted, world accepted nomenclature of naming organism. Naming organism is a world accepted formal method of naming an individual. So this was initiated or presented by Linnaeus. It's initiated by Linnaeus. He is a Swedish naturalist and he initiated binomial nomenclature. And this binomial nomenclature, that particular scientist initiated giving names by Latin, not in English, by Latin. Latin words, binomial nomenclature. Therefore, for example, for human, we give the binomial nomenclature. First part, that is the noun, homo, that is referring to the word man. So he's the person who introduced the nomenclature that we accept today. We call binomial nomenclature and lived from 1702 to 1778. So this is his very old or very first publication based on the naming of organisms. And if we take this naming introduced by this one, Linnaeus in Latin, has always two words for individual animal, two words. You can see always the first word is a type of a noun. And this noun, the word noun, sometimes describe some features of this particular thing. And you can see this noun, the first word, always initiate with the capital letter. Capital letter. Words are Latin, or we call italic. And the italic words, usually we write in italic form. Italic form, if not, if it is a printed version or handwritten one, we have to do it underline. Italic or underline. This first word noun follows with the second word, which describes further about this particular organism or individual. And this second word, the word initiate with a simple letter, and this is a description to the first word. And the first word, therefore, we refer as a generic name or genus. Generic name or a genus or the genus. The word initiate with capital letter and it is a noun. And the second word is the specific epithet. It is like adjective explanation of the first one. And the second word we refer it as to its species. So if we question to give the species name of this particular individual X species name, we have to write Homo sapiens. But if there's a question to write the generic name or the genus, you can only write this generic name, only this species name, all this. And when you are writing, you have to write it first letter capital and the whole letters should be italic. If not, in writing, you have to underline it. It is a formal thing you have to remember from your year 1 up to throughout your study in the university as well as after. So it is the English Roman letters, name initiate with capital letter generic word, and the species name do not have a capital letter. If it is writing, should underline the words, and also for one particular species, the name does not repeat for another species. Means two species do not get the same scientific name. Do not get the same scientific name. In addition, generic names we can abbreviate with initial. When we are writing continuously in the same paragraph, we can initiate with Homo sapien at the beginning, but when we are continuing, we can abbreviate the first word H dot, but not the species. Then we can refer the common name utilized for that particular species in the initial state. For example, if it is a house sparrow, Gayfurula house sparrow, we can write the scientific or zoological name like this. Again, this particular species, if found by an author, if one particular author has given the name for this particular species as Passer domesticus, we can write within bracket the author name with the year it is being found. For zoological words, later on, we can abbreviate again the author's name as capital first word, Linnaeus L. dot, if it is for zoological words. But if it is for botanical words, you have to write the name continue. And again, there may be sometimes species. After the species, we call it as subspecies. Subspecies for human subspecies, sapiens, the subspecies. Then, for example, imagine one particular person found this particular individual first time. Again, after sometimes the second person may describe the first individual with a different name without knowing that previously it is being identified. In that case, we have to write the year of its identification within brackets with the author's name, if it is David, within brackets the year 2021. Same individual identified by David in 2021 and given a name, different name. So if they come to know if it is the same species described by the second person, we can later improve it as The second naming given for the same individual previously as a synonym. Synonym, we can write it like this. Okay. For example, Passer domesticus in 2021 identified by David as Amaranthus, something like that. In that case, we can refer this as a synonym to the previous one. But Amaranthus is not the Passer's similar name, it is different plant. One I am giving you just an explanation. Now, what are we going to do if there is a new species identified by a person, new species, without knowing that it is being described previously? For example, while doing a sampling, you found an individual which is completely different and unable to classify according to the available classification. In that case, you can imagine that it is a new species. Imagination. But how are you going to do it? First, you have to describe with available classification and available identification guides whether it is being identified previous. If not, you have to designate a preserved specimen in a available national museum where it can be accessible by others for research because it is the only individual found firstly by no one else, only you. It is the deposition of the specimen identified in a museum. We call it as a holotype. So what is a holotype means? Is the specimen which is new to the world and going to be described. Until then, you deposit for others to access in a museum, holotype. Then you can propose an appropriate scientific name for this particular holotype. Propose an appropriate scientific name that you can decide basically based on the features available or based on the place where it is being found or based on the person who is identified. You can propose scientific name. That is left to the person who found it, but it should be binomial nomenclature. Italic names, two words. First is noun, second is adjective. Then you have to describe full information about the species you found, description, and you can write a full scientific documentary. Full scientific documentary, prepare and publish. How are you going to publish? It is either in the internationally accepted forum, conference, or printed journal publishing, so that everyone knows after referring to this particular publication, this particular species being found by so and given the name this and that. Then that has to be submitted to the scientific name of new species, scientific name, submit to the ICZN if it is zoology, that is International Code of Zoological Nomenclature for approval. If it is a botanical species, to be submitted to International Code of Botanical Nomenclature.